Hello, everyone. My name is Winnie Wong, and I am a trustee of the Dana-Farber Cancer Institute. I would like to extend a very warm welcome to our new and returning guests to the breakout session that covers progress in the GI Cancer Center. Because of the time limitations this year, we will forego the individual introductions, but your names are all on this slide. On behalf of the Institute, I would like to thank you for your past, present, and future support. In addition to financial contributions, many of you have given us sound advice and also have used your connections and networks to expand our reach into the community. For all these efforts, we are very grateful. Now, in case some of you have needs a little technical help with how to field questions, here's a few tips. We will start on the right-hand side of your screen. If you go to the bottom right panel where it says chat, you will see a box that has a question mark. So if you click on that anytime during the presentations, you can click on that question mark icon and it will turn green. Then start typing your questions in the box below that. These questions will then be queued up and our faculty will field these questions and address them each during the Q&A session. If you didn't have a chance to type in your questions during the presentations, you can also ask your questions live. On the top right of your screen, you will see a participant panel. Under your name, click on the raise hand panel, and then it will light up, and then we will field the questions as we go along. Okay, so feel free to use the Q&A box anytime, and the questions will get queued up. Now, if you have any questions, you can send a note to Angel and she will help you. Or if you need technical support, let us know. So let's see. Okay, the agenda today includes an overview by Brian Wolpin, who is director of the GI Cancer Center. There will be a Q&A session with the faculty, and then there will be two scientific presentations to tell us progress in GI cancers. This will be followed by another Q&A session before we wrap up and finish at 2:50 p.m. And then the session will go to the closing remarks by Laurie Glimcher, president of the Dana-Farber. So now we can proceed and kick off to... I think we can go on to the next. Yes, okay. So our first speaker is Brian Wolpin, who is chief of the division of GI oncology. He holds the Hale Chair in Pancreatic Cancer and director of the Hale Family Center for Pancreatic Cancer. Among his many leadership hats, Brian is also a well-renowned scientist in the field of prostate cancer. Dana-Farber has adopted a laboratory to clinic strategy in our fight against cancer, and Brian is a living embodiment of that approach. He's a world leader in his field, and his efforts in research have broad implications in the treatment, detection, and prevention of pancreatic cancer. So we will now have Brian speak. Thank you.
by just thanking you all for being here. We have a number of new members, and thank you for joining the group. And as Winnie said, thank you to any of our members who come back year upon year and have given us so much help over the years in building the program that we have today. I also want to thank Winnie for her leadership in designing and putting together this program. She's really an amazing partner with us to design and implement the program, and we are very thankful to her. And then obviously also thank you to Angel Kettlewell, who helped organize the program today. So to start with, this is the, if it will advance, and we can use that. So to start with today, here's a basic outline of the things that we'll talk about. So, we'll start off with what the mission of the center is. Those of you who have been here before know this, but I think it's always important to reiterate this. And we obviously do have new members too. We'll go through a little overview of the types of cancer that we treat and that we investigate within this center. This is a relatively unique center in that there are many types of cancer that fall underneath the GI cancer umbrella. And as you'll see, this is really one of the largest disease centers, both clinically and from a research perspective at the whole institute. And so we'll briefly review that. We'll talk a little bit about what the organizational structure of the GCC is. And whenever I say GCC, that really refers to the GI Cancer Center. So that's sort of the abbreviation that we use. So my apologies if I flip back and forth. And we'll show you a little bit about our volume in terms of patients, clinical volume, and how that's been affected by COVID. Then we'll talk a bit about people. I always like to give each year an update on new people who have joined the group. We've been very fortunate to year upon year grow the group. And that has in many ways been a direct output from all of your support, as the philanthropy that we raise is used to support new faculty. So we'll talk about that. And we'll talk then a bit about some brief research updates. Really, we use this to give you a bit of a sense in a more broader view of the work that's going on in the center. Both Nalay and Nadine are then going to give you a more detailed view of their particular research. But really, the amount of research going on in the center is so large. There's so much interesting work going on. I'd like to be able to give you brief sort of snippets of that so you have a sense. And then we'll talk a bit about the year in review and then needs for the future. Then we'll just reiterate again the agenda for the session with a question and answer session to follow this, and then talks for Nadine and Nalay. So to start with the mission. So I think most of you who have been here before are well aware of this. But we have a number of different things that we really use to guide us. And first and foremost is providing expert and compassionate care for patients and their families. And this has really been the main focus of the center for many years, really since the center began under Bob Mayer's directorship. Second, and equally as important, is the research that we do, right? And really this is the idea that while we're taking amazing care of the patients that are here now, we want to be the movers. We want to be the ones who make the care a year from now or 5 years from now better and different than it is now. And this is really one of the amazing strengths of the Institute as a whole, and I think the GCC really embodies that, is doing both of these things at the same time at an exceptionally high level. So both clinical care and research that goes together with that. And then we take very seriously our responsibility to educate the next generation of providers, both on the clinical side and scientists. And we try every year to highlight that aspect of what we do. And you'll also see one of the speakers today, Nalay, just started his lab a couple months ago after having come up through the Dana-Farber system. And he'll tell you about his work, but really an embodiment of this idea that you educate the next generation of smart people who are going to come help us fix these problems. And then the last is not to be too inward looking, right? We want to look beyond just the walls of Dana-Farber, and it's really our mission to help eradicate GI cancers around the world. So not just here in the United States as a whole, and then beyond that. And that really is an important aspect of what we do. So just to start off with, well, what are the types of cancer we deal with in this center? Because there are quite a number, as you can see here. So I made this joke last year, and my daughters would say it's a dad joke, but obviously the organs here are not that color in your body. They're just to mark off the organs that we take care of. But you can see here that we deal with everything from the top of the GI tract to the bottom, and everything that comes off of it, right? So there's a lot of kinds of cancer that we deal with. Esophagus cancer, stomach cancer, cancers of the small bowel, appendix, cancers of the colon, the rectum, the anus, and anything that comes off of the gastrointestinal tract, pancreas, gallbladder, bile ducts, liver. And then, as a number of people in the audience know, some from personal experience, there are also cells within the gastrointestinal tract that can cause neuroendocrine tumors. So we take care of patients with that. And then there's also cancers where doctors aren't really sure where they're coming from. They're somewhat mysterious, and those mysteries end up with us too. So those are sometimes called unknown primaries, where a cancer shows up but we're not sure where it came from, and we are the center that takes care of those people too. So you can see really a large number of patients fall under this umbrella of the GI Cancer Center. And if you want a sense of that from numbers, I think numbers only go so far, but they do sort of hammer home a point. And the point I really want to make here is we're one of only 13, right? So there are 13 disease centers within Dana-Farber, but we are among the cancers we treat, almost 30% of all cancer death is from our cancer types, right? These are common cancers and they're tough, right? These are difficult cancers. So 1 out of 13 disease centers, but 30% almost of all cancer death in the US, just in our single disease center. And you can see that's actually because a number of these cancers are really tough to deal with, right? So colorectal cancer is the second leading cause of cancer death in the United States. Pancreas is third, and what's called hepatobiliary or liver and biliary is fifth. And so again, these are common and difficult cancers, and ones that really need a concerted effort and a team-based approach to solve. And again, we so appreciate all of your support as we take on this big endeavor. So how do we structure then things? This is obviously a lot of different diseases in a big center, and I'll show you the people that make up the center. But the main way that we organize this is within 5 what are called subcenters, right? So we divide it by disease sites. So we have a subcenter that deals with colorectal cancer, one that deals with neuroendocrine tumors and carcinoid tumors, one that is for the liver, so liver cancer, another that deals with pancreas and biliary cancer, and then another that is for stomach and esophagus. And you can see each one of these subcenters has a set of leaders that really pushes home both clinical objectives and also research objectives. And then there are multiple faculty that sit within these subcenters. And it's really our goal to make sure we have a broad range of faculty, from bench scientists through what are called translational scientists, who are between the clinic and the bench, and clinical trialists, and then clinicians within each of these disease areas, so that we can make progress for our patients in each of these areas. And then you can see we have a number of other centers or programs within this. One which we heard about last year from Kimmy was the Young Onset Colorectal Center, which is housed underneath the Colorectal Cancer Center as a whole. Today we'll hear from Nadine, and she's going to talk a bit about some of her work in patient-reported outcomes data and how that affects patients, and also how she's then applying that to an older adult program. So patients who present at older ages have a number of unique challenges when they fight cancer, and Nadine is really amazingly poised to deal with that and help our patients thrive. And then you can see here we have other leadership related to the clinical operations of the center, related to the clinical trials we run, related to the translational work we do, which really means collecting tumors and blood samples, related to how we think about the genomics or the tumor DNA, which is something we call the GI Target Project. And then because of the fact that a number of our cancers are associated with Lynch syndrome, we also have a strong relationship with the Lynch Syndrome Center that's led by Sapna and Matt. And Matt is a GI oncologist also within the GCC. So you can see this is really how we handle the diversity and depth of work that we need to do by having a multitude of important leaders within the GCC that drive home the clinical work and the research. So next I wanted to move on to clinical volume. And I think those of you who have been here before are well aware of this sort of curve, which is that every year we see more patients and do more patient visits than the year before, and that's been going on for many years. You can see this straight line all the way back to 2005. And the only thing that's been able to bend this curve from its usual shape has been a global pandemic, right? And so you can see that this upcoming year, or the the past fiscal year, we did have a slight drop in the number of patient visits in this past 12 months. And that really, as I'll show in a second through a number of slides, is related to COVID. And I think there'll be more opportunities to ask questions about that along the way. And we'll also highlight a little bit of research around COVID that we've been doing. But really, only a global pandemic can slow down the number of patients who want to come to be seen in this center. We obviously wish there were fewer, but I think really the amazing care that our doctors provide are the reason, and our nurses and staff are the reason why the curve has looked like this for so long. I will also take this opportunity to say, if you have questions that you note along the way, please put them into the question box during the talk. I think that's often helpful for us to then be able to answer some of them at the end. So here's just a brief overview of what COVID has done. Right? So COVID has definitely had significant effects on all of us, certainly in many different parts of our lives, but it certainly has also had effects on our clinical volume and our ability to see patients. So you can see here, this is a chart put together by Steve Flaherty, who's the program manager or program director for GCC. And these are weekly, so each one of these bars is a week, going back 1 year, of the number of visits that we've done. Okay, so this is a weekly number of visits. You can always tell this one, the low one in the middle, is the week before Christmas and New Year's. That's always when we see the fewest number of patients. And so you can see that orange bar shows you sort of an average number of visits that we do in an average week. And then the blue arrow shows you right as COVID comes, right? So you can see immediately there's a decline, right? It's about a 10 to 20% decline in the number of visits that we were doing as we tried to keep people, safe and out of clinic when possible. And then you can see all these orange colors showing up instead of blue. And orange was a shift, and you can see a very rapid shift to trying to also do virtual visits, which you can see from before the blue arrow we had never done. And so that was not something that we did. It really wasn't until COVID that we started to do this. And now you can see there was actually quite a number of virtual visits. And now you can see as we went through the summer and now into the fall, our volume is about back to where it was, right? So the most recent data in September and October suggests that the total number of visits we're doing now has almost reached back to where it was prior to COVID. But there clearly is a multi-month period here where our volume is less, right? And we can see that also if we look a bit at the infusion room visits. So this is really financially where a lot of the funds for the disease center comes clinically, right, is giving the therapy to our patients. You can see that went down a little bit. There's a slight incline or decline really to the curve, but relatively modest. And that's because many of our patients with GI cancers are sick patients and they need to be treated, and that needed to continue even during COVID. Really what it required is a lot of safety measures to allow them to come to clinic, but they still needed to come to clinic for their treatment. And then the last piece I just wanted to show, this is our new patient volume. And I think this is really the metric we've been keeping the closest eye on, in particular of late. And as you can see, we do see a lot of patients who travel from far, right? And so coming into Boston, particularly at the height of when cases of COVID were highest in Boston, was difficult for people. And so you can see at the blue arrow, again, the gray bars are the in-person new patient visits. You can see we went from doing 80 to 100 new patient visits every week, which sort of was our normal volume, to falling off about 40% early on. That has slowly crept up, right? It has slowly increased again. It's about 90% currently of where it was before. And I think this is the one that's really going to determine for us next year, right? Because the number of new patients we see is then what determines the number of patients we follow and take care of going forward. So I think this is an important metric for us as patients come back. And in particular, trying to make sure, and a lot of energy and time has been spent trying to make sure that the environment is safe for patients to return to see us, right? Cancer is obviously a serious disease, and these patients need to be treated. We want to make sure they can come to do that with us and do so safely. So let's do a brief overview of people. I do like to do this every year to show you the faculty in the GI Cancer Center. So right now we have 29 full-time faculty. So these are all physicians who take care of patients and do research. Some of them may do more clinical time, some may spend more time doing research. And then we have a number at the current time, 4 instructors who are sort of making that transition from being in the lab to ultimately having their own lab to run. And so we have 4 instructors in that situation too. So 33 total faculty in the GI Cancer Center. Again, one of the largest disease centers at Dana-Farber. And in terms of new faculty, so many of these faculty we have hired in the past year prior to COVID, trying to get ready for when Chestnut Hill opens, right, which is our new facility that you've heard some about, and I'll highlight again in another slide or two. And also dealing with the fact that most years, non-COVID years, we see continued rise in our patient volume. And to take care of those patients, we need to make sure we have enough physicians, who are working along with enough nurses and staff to take care of these people. So just to highlight, the folks in red are the new oncologists that we've hired in the past year and have started. So if we start on the left, you can see CC started in January. She came to us from Stanford. She's both a medical oncologist and also does work in the genetics clinic, similar to if you heard the early detection and prevention seminar that was earlier this morning. She works in that setting too. So my slides just went back to the beginning. I'm going to just catch us back up to where we were here. Okay. So hopefully you can see this back now too. So the second is Ben Schlechter. So Ben came to us and started in December, and he came to us from the Beth Israel. And Ben is really interested in colorectal cancer and neuroendocrine tumor, and in particular running clinical trials to help find new therapies for these patients. The third that you'll see is Nalay, and Nalay is going to talk later today. So Nalay was a postdoctoral fellow in Adam Bass's lab within the GCC, did his fellowship here at Dana-Farber, and now has his own lab. And not to brag, because Nalay is very modest, but there was a large national search. So lab-based fellows from all over the country applied, and they could work in any cancer type, so not GI cancer specifically. And of all of those people who applied from all over the country, and actually there were even a couple international, Nalay won that search, meaning he was the top candidate and was offered this job. And we're just exceptionally excited that he is going to stay with us and continue to work here. And as you'll hear, is just doing some amazing work. So the next person on the list you'll see is a new faculty member who's going to start in January, so he hasn't started yet, is Harsh Singh. And Harsh actually is in the lab now. He also did his fellowship here working with Ramesh Shivdasani. And Harsh is really thinking a lot about why do cells become cancerous? And how does expression of genes within cells turn them into cancer? And so he's going to continue that work, but now as an attending and a full-time faculty in the group, which we're very excited about. And then the last you'll see there is Sarah Slater. And Sarah came to us from Mount Auburn. And Sarah sees patients in clinic, and her other interest is international health. And so she's traveled actually all over the world with partners in health, helping to set up cancer care in other countries, including in Africa and Haiti, in Eastern Europe and other places. And we're really thrilled. Sarah joined us in July. So these are the new people who are joining us this year. And I do always like to, although often we don't have people who are leaving, but if we do, just to note that, we do have a couple people who are going to leave the group this year. One is Adam Bass, and Adam talked last year, so you all know he does basic science research in gastroesophageal cancer. And Adam was really offered a large leadership position at Columbia University in New York. And we've talked before that some of the ways we use philanthropy is to try to retain the best faculty. And in particular, New York causes us trouble, right? New York, there's a lot of philanthropy in New York City. And oftentimes when we do have someone successfully recruited away, which isn't too often, but when it does happen, New York is often underlying that because of the money they have with philanthropy there. So again, just to reiterate how we use some of these philanthropy funds, we really use them to try to retain the best faculty and recruit the best faculty to be learning and practicing here, here at Dana-Farber. So, and the other is one of our lab-based instructors, Ankur, works in Adam's lab, and he's decided to go into industry rather than continuing his work in academia, and is going to actually join Novartis in a couple months. So those are the two people that will leave the group in this year. But as you can see, we've really also grown quite a lot this year with multiple new faculty joining us. And then I would really be remiss if I didn't highlight the amazing nurses who work in the GI Cancer Center. We couldn't take care of the patients like we do without them. And that also, we've seen a tremendous amount of growth in our nursing groups too. So you can see on the left what are called APPs, which are either NPs, so nurse practitioners, or physician assistants. We've hired several new APPs in the past year, again, because of our volume growth, really over the past couple years pre-COVID, really was starting to make us need more nurses and more PAs to help us care for these patients. And again, these people deserve a tremendous amount of thanks for the amazing care that we deliver. All right, so one more clinical slide, and then we'll talk a bit about some of the research updates. So as we know from last year, Chestnut Hill was scheduled to open, and Chestnut Hill is a new clinical facility that we from the Longwood campus will go out to staff. So it's all of the same doctors that are here in Longwood, but will also practice in Chestnut Hill. Was scheduled to open last month in September. Because of COVID, that got delayed, and that now will open in January. So we are really in the thick of a lot of planning to try to make that transition. It's a big transition for us. We have 40 sessions, meaning 40 half-day sessions of clinic a week there. So a lot of patients will be seen there. And we're in the process now of trying to make sure that's a smooth transition, both for the patients, which we want to make sure of, and also for all our staff and physicians and nurses too. So this is really the main clinical change that will happen in the next year that we're planning for currently. All right, so I think what I'll do is I'll just do some brief research updates across the different cancer types. Again, not so much to go into tremendous detail, but more to give you a sense of the breadth of work that's going on in the disease center. And that really spans everywhere from very basic bench work, trying to understand the biology of cancer, through to new treatments. And I hope you'll get a good sense of that from these studies. So to start off with, I've chosen 2 studies from each disease. They're all very recent, so things that have been published very recently. The disease center is very, very active on the research side. There are so many publications. You could choose from really dozens and dozens every year. So I apologize for ones I didn't pick, but I really tried to choose ones that were very recent, that have just come out, to give you sort of the latest science in terms of what we've been doing. The first one on the left is really what Nalay is going to go into a lot of detail on in his talk, but I did want to highlight this. It's a study that he did with Adam in the laboratory, really trying to think about new mouse models to study gastric and esophageal cancer, and thinking beyond what other people had done before, and really creating new and original ways to study this disease. And that, I think, has provided a lot of very interesting insights into why these cancers develop, and then how we can treat them better. And Nalay is going to talk about that in more detail. And then I highlight on the right a study from Peter Enzinger that was published just last month. And this is really the tip of the iceberg, but it is the implementation of immunotherapy in gastric and esophageal cancer. And this has really now been a series of studies, many of which Peter has been directly involved in, and that we've enrolled patients to here at Dana-Farber, to move immunotherapy into treatment of gastroesophageal cancer. And we've now started to see some real successes with that in this past year or two. And Peter has really done a lot of work to help highlight or to help move that work forward. So for some reason, it keeps going back to the beginning. Let's go back. All right, so that was gastroesophageal. For colorectal, I wanted to highlight a couple studies that, again, I think show the breadth of work that's going on. So the one on the left is from Jonathan Nowak, who's a pathologist who works within the GCC, and Marios Giannakis, who's a medical oncologist in the GCC who also has his own laboratory. And what they wanted to do is say, we do a lot of resections for colon cancer, right? Someone presents with a colon cancer, and we take out the tumor, right? The surgeons do. And then we look at it under the microscope. And the idea of this study was that there's a lot more information that we could gather from studying these tumors than what we currently do. And that information should be helpful to us to tell us what sort of treatments people may need, or whether they maybe don't need treatment at all. Maybe the surgery alone was sufficient to cure their cancer. And so what Jonathan and Marios did is start to leverage machine learning, right? So taking this idea that computers may be able to see patterns in the way immune cells are infiltrating tumors to help us understand the biology of the cancer and how we can treat it better. And this is really one of the first efforts nationally to do this. I think a really interesting study, and the figure there starts to show how the computer, with help from pathologists, can help start to mark different cell types, different cell types in the tumor, and then understand what that means for the biology of the cancer. I was also on the panel this morning, and I think my voice is starting to feel a little worse for the wear. Give me 1 second. All right, and then the study on the right is a number of studies that Kimmy and Jeff have led, trying to understand what are factors that patients can do to help treatment work better or prevent cancer from coming back. And this has included things like physical activity and diet, or in this study, coffee intake. And they've done a real amazing job cataloging these different things so that we in clinic can provide recommendations to patients on lifestyle factors and ways that they can take control of their care, in addition to some of the therapies we may give them. And this was a paper they published on this that came out just last month. And then I wanted to briefly highlight a couple studies in neuroendocrine tumor. So to the left is a paper from Jen. You can see Jen Chan, which came out in August. And to the right from Ramesh Shivdasani, that came out in September. Again, you don't have to go back very far to find influential work. Even much of this work is really just the past few months. But this group is so productive and really does so much research that there's constantly discoveries being made. But you can see on the left is a committee that Jen chaired in order to come up with national guidelines for how to treat pancreatic neuroendocrine tumors. And really, these guidelines are the guidelines everyone, not just nationally, but really internationally, use to help decide how to treat our patients. And again, not to brag a bit, but this is really Jen chairing this entire committee to come up to, to come up with recommendations that everyone around the world can follow. Again, just shows the reach of Dana-Farber is not just internal, it really goes way beyond this, way beyond just our internal work, but to how people are caring for patients with cancers across the nation and across the world. And then the study on the right is from Ramesh Shivdasani, and this is really a study that's looking to try to understand how cells in the intestines decide what kind of cells they should be, and how do they interact with one another to do that. And if you understand that, if you understand some of those basic mechanisms of how cells decide their cell fate, that actually informs you a lot then on why they become cancer and how we may be able to prevent that. So a really interesting study from Ramesh and his group. All right, and then I'll finish with a couple in pancreas cancer. Again, this is always so hard. We have so much work going on, and we obviously have a number of diseases. And just trying to give you a catalog of this is not easy, but just to give you, again, a flavor of things. So you heard, if you were at the committee meeting several years ago, Andy Aguirre is one of our laboratory-based investigators who studies pancreatic cancer. He also trained at Dana-Farber and then stayed to run his own laboratory here, which he now does. And this is a study he did that made an observation that the way we decide what drugs may be useful in patients are often determined by studies we do in the lab with what are called cell lines, which are these cancer cells that we include in studies in the lab and try to treat with drugs. And if it works in the lab, well, then maybe we should try it in people. But this is a very inefficient and labor-intensive process. And so Andy designed an approach to multiplex this, to do this across many cell lines at once, using a barcoding system that's really quite novel and interesting. And what this does is it allows us to start to test drugs much more quickly, and then try to make decisions more quickly about whether those drugs are things we should be testing in people. And so it's really an approach to try to accelerate the time it takes to decide what things seem to be interesting in the lab so that we can then move that into helping people. And again, really thinking that the faster we get new medicines to patients, the better things will be. And this study is actually a study from my group where we're trying to understand mixtures of factors that may increase risk for pancreatic cancer that could then allow early detection and screening. And this particularly was focused on people who developed diabetes and had changes in their weight and whether they were trying to lose weight or not. And this started to help us tease out who might be at risk for this disease and how might we then consider them for screening. So the last research effort I'll mention is not actually GI cancer focused. It's actually COVID focused. So I think as some people on the video know, Jeff Meyerhart isn't only part of the GCC, he also runs the entire clinical trials operation for all of Dana-Farber Harvard Cancer Center. Really a tremendous responsibility and effort. And so when COVID came and we needed to start to study COVID and how it was affecting our providers and staff, the Institute really turned to Jeff to help lead this effort. And so Jeff is leading a large study to understand exposure and symptoms to COVID and how we can start to use that information to make sure we keep our staff safe and ultimately keep our patients safe. And again, this is a really important thing for us in the coming 6 months, as COVID has not seemed to go away and really continues to be an issue for us. All right, so we'll do a slight year in a review, and then we'll talk a little bit about our goals for the next year, and then we'll open it up for some questions. And then obviously we'll move forward with 2 more talks, one from Nalay and one from Nadine. So if I had to summarize this year, certainly we didn't see COVID coming, and that ended up taking quite a bit of our effort and time to navigate. But really what I would stress is the clinical care never suffered. It really was continuing to be the most amazing clinical care from our providers, our nurses, our staff. It really kept the same standards of excellence that we have always maintained, and that will continue regardless of what COVID does in the months ahead. And then I think really for us in the coming year, it's making sure in the midst of COVID that we also, in a very safe and structured way, transition to also staffing additional center, that's about 4 miles from our Longwood campus. I would say we continue to grow. We continue to hire new faculty with new expertise. We're going to continue to push the envelope and move our understanding of cancer forward and improve the way we take care of our patients. And I mentioned these people by name before, but Ben and Cece and Sarah and Malay and Harsh, all coming to us within the past year to continue their work. And we also, as I also mentioned, have added now multiple new nurses and nurse practitioners. As again, our clinical care operation grows, they really are such a critical component of how we take care of patients. And we need to make sure we're adequately staffed to do that. And then you can see, hopefully I gave you the whirlwind tour a little bit, but some impression of just how deep the work that goes on here is, just the incredible breadth and depth of the science. And that really is what allows us to change the care for our patients, is doing the science both in the lab, in the clinic, and in between that moves care forward. And it really is the major commitment that we've made. And I think you can see from the people we've hired, again, a diversity of interests and really covering the whole gamut of the different cancers and biology we need to understand. And then another piece to this is really ensuring we have options for our patients to try new things, right? To have clinical trials. And that's really a major focus of what we do, and that continues. And then last, it's hard to convey this to you, particularly not in person, but the faculty that we have here are really the national and the international leaders in GI cancer. And it is amazing. If I had more time and Angel let me have more slides, which she would not, but if she did, and I could list all of the national and international committees people serve on, all of the national and international consortia that they run, all of the research efforts that they do that really lead the nation, it would just go on for slides upon slides. I mean, it is really an incredibly distinguished faculty that is really replicated nowhere else. And I think the thank you at the bottom is really because this is a partnership, right? You all help make this happen, right? And it's the support you provide. Some of it is obviously monetary through philanthropic support. Some of it is the support that you provide in terms of outreach to other people, and also to to ensuring that we have good ideas going forward from outside our own ranks. So I really do thank you all for your efforts. All right, so these are basic needs, and I can see that Angel's telling me I'm running over, which I usually do. So I'm going to try to do this fast, But what I would say is, we talked last year, recruitment of lab investigators remains a really major priority. We have a lot of cancers that we need to figure out within the GI Cancer Center, and a lot of that really starts from basic work. And when you have an opportunity to tell Dr. Glimcher and others at Dana-Farber, what does the GI cancer need? This is one of the things that you should say. And it also is what helps with your philanthropy, right? When we recruit, or in some instances try to retain faculty, that's what we use philanthropy for. And so this is really important to us. Another, and this is something I've talked actually in particular with a couple members of the committee, is once faculty are here, how do we make sure they have the resources they need and make sure that they have the ability to do the work that we want them to do? And so sometimes endowed either chairs or investigatorships allow us to do that. And so thinking about how we continue to fund our investigators. And then quickly, we do have a number of very high priority projects that are moving through. I would note again, Nadine is going to talk about her older adult program. I'm very excited for that. I think that's a very important program for the disease center. Bioinformatics and data science, big data ideas, again, really important. And then how do we bring teams together to solve these cancers? These are complicated. We need funds that we can use to bring teams together. And there are clear examples of that in the GCC, whether it's the Hale Center for Pancreas Cancer or GIFT from Russell Grusso for colorectal cancer. We have really been able to leverage those funds to bring large teams together. And then I'll finish by saying, we really need your help to stay connected with our supporters, right? It is a tough time, and not as many people can give as maybe could give in the past. And a lot of the events that we use to raise funds have been either virtual or haven't raised as much money as they normally do. And these are things that we need your help with to make sure that we continue to do the work that we're also eager to do. All right, so I will finish. So I think what we'll do, here's the rest of the agenda for today. So you heard from me and survived that. We'll do a brief question and answer now. And that will involve myself and then also the other faculty who are here. So just briefly, Jennifer Chan, which a number of folks here know, directs our Neuroendocrine Tumor Center within the GCC and is also the clinical director of the GCC. So working very hard with myself and with Steve Flaherty on how we deal with our clinic during COVID and also move to Chestnut Hill. Bob Mayer, I think most people here likely know, was the past director of the GCC, is currently the Dean of Admissions at Harvard Medical School, and is the one who trained most of us who are on this call, myself included. The next is Nadine Mccleary, and we'll hear from Nadine and some of the work that she's been doing, including the older adult program, and also some of the work she does with patient-reported data. Jeff Meyerhart, which a number of folks on this call also know, helps run the Colorectal Cancer Center within the GCC, and also, as I said, really runs the entire clinical trials operation for the whole institute. And then Nalay Sethi, who will also present and is one of our new lab investigators. So that was a bit of a mouthful. I'm sorry I took us through so fast. Let me take a look and see here what questions we have. And again, it's trying to cram in a lot of information, and please put your questions in the question and answer box so we can answer them. So here is a question, and it is about one of these questions, what is the status of liquid biopsies in GI cancer? So meaning how we use markers of the tumor in the blood to help with treatment of GI cancers. So maybe why don't I actually send that one a little bit to Nalay to start with. I know some of his work has had some relevance to that, and then I'll ask another of our panelists too. But maybe Nalay, do you want to give a brief sense of that?
Sure. So liquid biopsies is something that has been pioneered in all cancers and used and entering the GI cancer space as well. The two main areas where we are thinking about liquid biopsies is in early detection and diagnosis, sometimes even of pre-cancer before the cancer has arrived. Sometimes we can see the molecular problems display itself in the blood. And then another area in which it's being used is when cancer has already been diagnosed and on treatment. Sometimes we can monitor the response of treatment through markers in the blood rather than having to continue to look at the tumor directly. So these are the two real areas that are still, I would say, in the early phases, but rapidly moving towards the clinic as potentially standard of care soon.
Thanks. Jeff, maybe I'll ask you too. How are you using this in your clinical practice? There's some data around colon cancer now to use these. How do you do that?
Yeah. So again, there's multiple settings where you can think about it. Is to use it like in a patient where you may not be able to get an adequate biopsy to be able to get the molecular profile of the patient. For a patient who's progressed on therapy, to think, are there new mutations that have come about? There's a lot of interest to follow people after diagnosis and after treatment who are without evidence of disease and to follow them as an early detection of recurrence. Some of those are just really, we're just not there yet. And there are some national trials which we're participating in. There's one specifically for stage 2. There's one being developed for pancreas cancer that Irini Pexais, who's at the Farber, is working with, again, the group through the network that we use on the, with the NCI. There's a particular one for colon cancer that's within the Dana-Farber Cancer Center that Marios Giannakos is working with researchers at the MGH, specifically looking at stage 2 colon cancer and thinking about if you have certain mutations, maybe additional therapy after if there's certain molecular alterations. So again, from that point of view, there's still a lot to learn in terms of how we can utilize that. And we have several projects that are ongoing to kind of figure out the role in those settings.
Yeah, thanks, Jeff. So I think I agree with those comments. There are a number of places it can be used, and I think we're just now integrating this into our treatment of patients, and then there's also trying to use it in early detection. Again, this could be a long discussion because this is really some interesting science and work, but I think we'll keep moving through people's questions. So another one is, how does supporting young investigators affect their research within the GCC? So maybe, Bob, I'll ask you, in as many years being a mentor to so many of us, how does philanthropy and support help investigators and clinicians through their career in that sort of support?
Yeah, it's a very important question. I think one of the unique things about training at the Dana-Farber Cancer Institute is the opportunity to become an academic leader. And that leader could be as a clinician, a scientist, a translational investigator, but that is really only possible if the institution can provide the support to allow people the free time to learn to do research, to network, to meet people in other institutions, to create the collaborations, just as Jeff Meyerhart was just mentioning with the liquid biopsies. And the funding that goes to young investigators is absolutely essential. Our national organization, the American Society of Clinical Oncology, provides 60, 70 young investigator awards every year. For us at Dana-Farber to be able to increase our number and even to endow them would be a godsend beyond belief.
Yeah. Thank you, Bob. I really agree. I think, there's the first step of getting the faculty here, right? So recruiting people to come here are really the stars, right? And that's one component of this. But once they're here, they need to have the funds and the freedom to do the work that's really going to be transformative, right? That's going to change the way we treat and think about cancer. And I think having both the funding support and also the moral support of having a team behind you who is really interested in your success makes a tremendous difference for young investigators. I wonder, Jen, if you might mention, you run so many interesting trials in neuroendocrine tumor, which is, not the most common cancer. How do you do then some of the additional work? Do you use philanthropy to help support some of what you do in a trial? How does that work for you?
Yeah, I think philanthropy is really critical, especially for rare diseases where there is not as much, for instance, foundation funding or NCI funding. So much of the translational work or, a lot of our basic lab research is funded largely in part from philanthropy. So it becomes quite critical.
Right. Again, I would.
Could I add something too?
Please,
I think if you asked the question to Laurie Glemcher, her response would be that she wants to be and is the president of an organization whereby if you have a good idea, the organization will find the funding and allow that to happen. That's what we saw with Kimmy and young adults with colon cancer. We're going to hear from Nadine about that in a few moments. That is what this place is all about and what really distinguishes it from other places.
Yeah, thank you, Bob. And I really couldn't agree more. I mean, I think the way that these efforts get started and that these new initiatives begin is one, a great idea from a faculty member, in that case, Nadine and Kimmy, and then the support to do it, right? And as Nadine and Kimmy could both comment, it's not easy to fund this work. We know it's important, but there's only a limited number of places you can go to get that support. And so I think having the internal support makes all the difference in launching these types of programs. All right, so I know, Angel, I know we're a little bit late, so we need to keep going. So we will have another question and answer session after both Nalay and Nadine talk. So think of your questions, keep putting them in the question and answer box, and we'll try to revisit them. And in my own defense, we are at a shorter time interval this year than usual, although I always run over, so that's not really much of a defense. But so what we have next, so Nalay's going to talk next. So I think I've mentioned Nalay a couple times now. Really just a brilliant young scientist who studied at Dana-Farber clinically for his fellowship and then worked in Adam Bass's lab, one of the other members of the GCC. And now, because he's done so much amazing work, as I talked about before, won a national search to open his own lab here, which started a few months ago. And I've just been so impressed of the work he's doing and the way he thinks about cancer and science. I really wanted him to come talk to you. And I also just wanted you, again, to see a direct example where we can recruit new faculty because we have philanthropy to get the best people here. So, Nalay, thank you so much for joining us.
Thank you, Brian. And I'm honored and grateful for all the comments that were said. I hope I can live up to those expectations. And in general, I'm grateful to all the members that are participating and supporting the GCC. It is instrumental. And since I'm talking now, I can also chime in on the last question about where philanthropic funds have been instrumental for me personally as a young investigator. As many people who are starting labs will share, there's a momentum that you need to build. You need to hire people, you need to get equipment, and you're using a lot of your startup funds to do that. But as a young investigator, I also have a lot of ideas, and I really want to push forward on those ideas without any delay. And for those reasons, I have even tapped into some of the philanthropic funds I already have to push ideas forward before I can get grant support from the National Cancer Institute or the NIH to support those projects. So in a very specific way, during a young investigator's career, I think philanthropic funds can galvanize new, exciting ideas before they get sort of traditional grant support mechanisms. So with that transition, I'm really excited to share some of the work I've been doing in the laboratory. This work started in 2015 when I joined Adam Bass's laboratory, who's been a terrific mentor of mine. And Brian Wolpin has seen this work throughout and supported it in its early stages all the way to now, where we are thinking about ways of bringing some of the new understanding into the GCC clinic and beyond. So I actually trained in breast cancer oncology during my graduate studies, but this slide is going to share with you one of the major motivations for me to move over to GI cancers. And it is that these cancers are, among the most deadly worldwide. I know Brian shared with you the statistics in the United States. These are the statistics from the world. Esophagus cancer accounts for the 6th most cancer deaths worldwide. Stomach cancer accounts for the 3rd most cancer deaths worldwide and has been growing in incidence. Excuse me if you see gastric on the slides, it's basically stomach cancer, and I'll try to use that in my words. And then finally, as everyone knows, colon cancer is one of the largest burdens of cancer deaths worldwide, second most common, and with this increasing rate in the younger populations that Kimmy is focused on and Marios is thinking about, and I too am thinking about how we can try to intervene and change the trajectory of this cancer for those patients. So these are the three cancers I focus on in the laboratory, and I think if I can remember correctly, fourth or fifth worldwide is liver cancer. So we are making up a huge burden as in the GCC. We're making up a huge burden of the total cancer deaths worldwide. And so why are these cancers so difficult to treat? Some of it stems from the fact that they're often advanced at the stage of diagnosis. This is somewhat to do with where they're located in our body and how there's not a lot of alarm symptoms that come from it. But I think even a bigger component is that these are very aggressive cancers, and they don't delay in their progression from something that is early to something that is late and can be consequential to the patient. When we do diagnose it and start to treat these cancers, they behave in a very chemo-resistant fashion. The therapies we give respond for a period of time, but then ultimately become resistant to the treatment. And part of the reason of that resistance is the diversity that these cancers display in their molecular profiles. Even if you're killing a subpopulation with your therapy of choice, there's another resistant population that emerges. And so this heterogeneity contributes to the fitness of the cancer. And these are the reasons why many people succumb to this every year. So my approach, and this is something that's cultivated way back to my graduate school days and been iterated on throughout my postdoctoral training, and is a philosophy and mantra for my laboratory, is this three-pronged approach. It's a simplistic approach. There's some nuances that are probably not captured in here, but in large part, I like to think about the cancer genomics, which is the human molecular profiles from human patients, and use that information to design the disease models and experimental systems that I can then use to achieve the rigor that we need to learn about the causal events in cancer. And finally, going back and forth between these two, we are able to shed light on therapeutic vulnerabilities and use that information then to go back, test it in our disease models, and hopefully come full circle back to the patient and see if there's efficacy in the treatment of GI cancers. So the rest of the talk is really going to be showing how I apply this three-prong approach in the study that Brian shared with you earlier. But this study actually started well before from a different study where we really wanted to focus on the human disease. And we decided to look at stomach, esophagus, and colon cancer, and dove deep into the molecular profiles across 6 different platforms. 3 platforms that are looking at the DNA, 2 platforms that are looking at the RNA, and 1 platform that is looking at the protein. And we took that information for 921 patients. So you can imagine this is a large amount of data that we were able to look at and then analyze carefully and try to make relationships that tell us how are these 3 cancers in common and how do they behave differently and what can we learn about them so that we can then come up with new strategies to treat them. And so before I give you one piece of data out of the many that we learned from that study, I wanted to share with you the 2 main ways. Again, this is a very simplistic explanation, but 2 main ways cancer alters its DNA. One is it can select for a mutation that it likes, which randomly occurred and allows the cancer to have an advantage. And the other way is by not mutating the DNA, but allowing for certain regions of the DNA to duplicate itself or even amplify it to greater fold. And this is liked by the cancer. And the yang to that yin is that if there's a barrier to this cancer survival or growth, they can then select for a deletion that loses something that's blocking its path towards cancer. So these are the main mechanisms by which cancer alters its DNA. And when we look at the second form, this amplification and deletions, we first noticed a distinct pattern in esophagus and stomach cancers, which I will refer to as upper GI cancers, when we compared them to the lower GI cancers. So this piece of data really shows you each row represents an individual patient, and each column is a specific chromosome in each patient. And you can see the red areas are amplifications and the blue areas are deletions. And from this bird's eye view, we notice that there's more granularity to the upper GI cancers and something akin to a Seurat painting. And this gave us a sentinel clue as to what could be an early event that leads to this sort of more finer granular copy number changes. And without giving you all the details of how we arrived to this conclusion, P53 was a key gene that we learned, unlike in the lower GI tract, is mutated early in the upper GI tract. And this very much informed my ability to design a mouse model that incorporates this understanding. The other thing I noticed, and this is a bit daunting, is that all these smaller amplifications and deletions are occurring throughout the genome. We don't know which ones are selected for on purpose and are actually driving the cancer, and which ones are passenger events that don't really have any consequence for the cancer. These actually, the passenger events make up majority of what we're seeing in this piece of data. So to me, I relate it to a shattered piece of glass that we are trying to now piece together. And I found that very daunting and very difficult. And this is why you can use patient data to help you get certain ideas, but then translating those that understanding into a carefully designed model is going to be the next step. And so I will start to introduce the disease model that we use, but in order to accomplish what I will describe with you in a little bit, requires a team of people, and I'm grateful and truly blessed working with a multidisciplinary team of talented people, from mouse geneticists to pathologists to computational biologists. So together, what we did was we thought, okay, P53 is an early event, but now what approach do we want to use to design a model that captures stomach cancer? And what you'll see that I'll describe is really stomach pre-cancer. We don't capture these in the traditional models. So the traditional models use other alterations that we have characterized in stomach cancer. We added on top of the P53, and in a very short latency, maybe a few months or a few weeks, you can get cancer. However, this to me didn't strike me as the most human-like approach to cancer development. Instead, cancer evolves. After P53 is lost, there's a period of time under which there's evolution and selection for things that the cancer likes. So piling on mutations did not, to me, strike as the best approach. But it is a feasible approach. It is a quicker approach, and I can understand why researchers utilize this, and I think we've learned a lot from this approach. That being said, I wanted to try to incorporate some of my medical background where we understand the risk factors that predispose to stomach cancer and try to incorporate that into a model. The pitfalls of this are that it takes a much longer period of time and much more money and resources that we need to invest into these types of systems. But for me, being here at Dana-Farber, having those funds available through philanthropic and grant support really made that second approach more possible than if I was someplace else. And so what are the risk factors for stomach cancer that we use? Bile acid is part of refluxate, and I thought about this as something that causes inflammation, and we know patients who have greater reflux disease have a greater incidence of esophagus and stomach disease. And then the other component was dietary carcinogens. Now, a lot of these carcinogenic agents that cause DNA damage are found in our body, and they're at very low levels, and our body can repair them, the alterations that are caused by it, or those cells die. But a constant and repeated dosage of carcinogenic agents is a risk factor for the development of these cancers. So I thought to incorporate both of these with early P53 mutations to develop an integrative mouse model. And so that's what's novel about this model system. And as you can see, this is how we administered those two risk factors to the mice over a year it took for these cancers to develop. And really, we were studying the pre-cancer. This was another advantage of the system that I found attractive. Rather than trying to create the shattered piece of glass, maybe we see the first few breaks by studying the pre-cancer state, and that allows us to understand what are the critical early events that lead to cancer evolution. So many models did not work, and seasons changed, gray hairs developed, but finally we got a model that worked, and this was the result. When being exposed to these risk factors, about 1 in every 5 mice got a precancer lesion in the stomach if it was normal for p53, but that rate jumped up by 3.5-fold to now 3 in every 5 mice were now developing these precancer stomach lesions. And so we spent many years studying this model system. This is just the early finding, but we then wanted to study it. And I'm just going to share one finding that really was consequential and influential in coming up with a neurotherapy for stomach cancers. And so there was a lot of unbiased analysis of the model system that helped us come to this result. But I want to share with you one of the more easier ways to visualize it. And actually, you're looking at right now the stomachs of mice that were part of these experiments. And what we saw was that first, you can notice that when p53 was being labeled by GFP, there was a greater burden and expansion of those cells compared to if p53 was normal. Finally, we noticed that the cell cycle, the mechanism of the cell that tells it to stop dividing, was activated. It was actually telling the cell to stop dividing because there's a lot of things going wrong when p53 is lost, and we saw the DNA damage. And that's where we saw that this break, the cell cycle break, was activated only where p53 was being lost and not really activated when p53 was wild type. And you can see that when you merge these two images together. So with that disease model understanding, I always want to go back to the human data and say, is this really happening in humans? And I'm going to move a little bit faster now, but we did notice it. We saw patients who had p53 mutations but did not have any alteration in that p16, which is a cell cycle break. We saw upregulation of p16. But then we also noticed that in the cancers themselves, some of them, you're losing p16. So the cancer is figuring out a way to overcome this break, this alarm signal that's being activated when p53 is lost. It now found a way to circumvent it, and that way was, exactly like I shared with you, deleting the gene altogether. So it was eliminated, and the cancer could progress past this checkpoint. And using that understanding, we then said, and defining this new subgroup of stomach cancers that have both p53 and p16 loss, we know there's an advantage to that. Is there any therapeutic vulnerabilities that have been revealed? And again, I'm not sharing the in-between research that really led to this discovery, but we found that a third break was activated. 2 breaks were lost in p53 and p16. A third break, which is called the DNA damage response, said, hey, wait, cell, we don't want to divide any further. But now, if we use an inhibitor that took away that third break, we saw that the cancer cells, stomach cell lines that had both p53 and p16 loss, which are denoted in red rectangles here, were more sensitive to these inhibitors than the other stomach cancers. So by releasing this third break, we took this train that's already losing, is already a runaway train. We took it completely off the rails, and these cells started to die. And we then went from the human data, which this is all human data, back to the mouse model, took these stomach units derived from these year-long experiments and asked, if you have both p53 and p16 loss, do you now have a selective sensitivity to these drugs, these inhibitors of the DNA damage response? And the answer was yes. So this is summarized here in the schematic, where we took a mouse model that was based on human data, human understandings. We took it all the way through to then learn about intermediate steps and ultimately cancer evolution. But that knowledge was instrumental in finding a newer agent that we can now push forward on the preclinical side with the help of the GCC to build a preclinical program and then ultimately a clinical program around. And that's where we are headed in the future. So this was a first study I shared with you, looking at the pan-cancer analysis, I then shared with you the integrative mouse model, but there are other portions of my lab that have focused on stomach cancers and learning about the hypoxia pathway being activated in these cancers and a vulnerability. And then I'm a burgeoning effort in the lab, and areas that I'm really excited about are actually in colon cancer. We're making great strides here, and I don't have time to share that work with you today, but very much happy for any of you to reach out to me through the Philanthropy program, and I can talk to you about this research at great length, as some of my colleagues would say. And I absolutely cannot end this talk without thanking all the people who have not only supported my growth, but supported the lab, my lab, Adam Bass's lab, the whole GCC. I'm only mentioning here Brian and James, James Cleary, but the whole GCC has been tremendous in supporting my work. And then the funding is the only way this work is possible. Both grant support and philanthropic support have supported the work that I shared with you. So some of you are part of the results that I just shared with you, and I really am grateful and appreciative of that. Thank you.
All right. Thank you, Nalay, so much for that. I think hopefully everyone gets a sense. This is a long-term labor of love to understand the cancer. Right? This is a multi-year project that he just showed you and talked about, but it really takes all the way from the beginning a basic understanding in the laboratory of how these genes are working and how they interact with one another to ultimately lead to what we think is going to be a new treatment approach for patients with gastric cancer or stomach cancer, as Nalay said. So it really is how new ideas form and then how they're carried forward that then leads to improvements for our patients. So thank you, Nalay, so much. So please keep putting your questions in the question box. I think what we're going to do is go directly to Nadine's talk, and then we'll circle back and answer those questions after Nadine speaks. So, we often try to balance the speakers that we have each year. So we often have a younger investigator and a more senior investigator. We try to do some work that's more laboratory-based and some that's more in the clinic. So Nadine's going to tell you a bit about some clinical programs and research that she's been doing around how you give patients more of a direct voice in their care, and you directly solicit their input as they're going through treatment. And then, as we mentioned before, this is then being incorporated in an older adult program that Nadine is leading. So thank you so much, Nadine, for coming to talk to us about this important work.
Thank you all for having me. And I just wanted to echo the comments of Brian and Nalay and other colleagues in expressing my sincere gratitude for the philanthropy that everyone here and many others have contributed to the GCC. Today, I wanted to start by sharing a bit about the patient-reported data program at Dana-Farber Cancer Institute and how that reflects the cancer care journey. And really that centers on a dear patient of mine who agreed to allow me to share her story today. So at the young age of late 30s, Amy was just starting a new life, a new journey, a new beginning, and really wanted to embark on a young professional life as a graduate of Assumption College, a new wife and a new mother, and starting a new job coincidentally with our partner's eCare program, which was at that time launching a new electronic medical record system called Epic. And in the midst of all of these exciting transitions, Amy was faced with also another new finding, a cancer diagnosis. And so I met her in the face of all of these transitions and hope, very concerned about what this cancer diagnosis of colon cancer meant for her life. And we talked about a diagnosis, we talked about treatment options, we talked about prognosis, but more practically, Amy wanted to understand, well, what does this mean for my day-to-day life? So how will I navigate my care? How will I feel while I'm on the treatment that you're describing to me? How can I cover the expenses of this care? How long will it take for me to recover from treatment? I have a newborn daughter. Will I be able to take care of her? Can I still travel? Can I still do things? Can I still be active? And many of those questions are not covered in our clinical trials. And in fact, we are missing a lot about the experience of our patients as we go through treatment. And so the patient-reported data program was really born in direct response to the needs of our patients, not only here at Dana-Farber, but across the US and internationally. So we wanted to document any report of a patient's status that comes directly from our patients without interpretation or reinterpretation by a clinician or anyone else. And so patient-reported data is really an umbrella term that encompasses most traditionally symptoms or toxicities from treatment, but also history, what has happened in the past, what's happened within your family, adverse events related to treatment, screening and early detection, and values and preferences. And so it's within this umbrella term that we started to learn about the benefits of documenting and really paying attention to what our patients, with their own voice, have to tell us about their care. There was a large study that was conducted at a sister institution by one of our faculty members here, Deb Schrag, and a colleague, Ethan Bosch. And what they found is of 800 patients diagnosed with advanced cancer, many of whom were computer naive and did not have an email address at the time that they embarked on the study, patients reporting on 12 common symptoms like nausea, vomiting, diarrhea, etc., experienced longer survival with fewer interruptions to their cancer care. So less emergency department visits, less hospitalizations. They were able to remain on treatment longer. And because of this, we were also able to see that patients engaged in this program with little to no impact on how the clinic operated. So we anticipated that maybe phone calls would increase, anxiety would increase, but it did not. In fact, this effort has been widely endorsed by multiple agencies, including agencies in Europe and here in the US, the National Cancer Institute, the Centers for Medicare and Medicaid, and the Food and Drug Administration. In fact, in 2009, the Food and Drug Administration endorsed that patient-reported data should be included as a part of any drug approvals, documentation, and application moving forward. And it wasn't until 2020, so 11 years later, that we begin to see this information incorporated into clinical trials. And here I'm sharing with you one of the last conferences I was able to attend in person. This was January of this year at the ASCO GI conference in San Francisco, where the IMbrave 150 study was shared. And here the investigators examined 2 drugs versus 1 drug for liver cancer. And included in the endpoints, the key features that the investigators were trying to understand, they included patient-reported outcomes. They included time to deterioration of quality of life. So how long will I feel well before I don't feel well? And also effect on functioning and time to deterioration or onset of symptoms. And what the investigators found is that patients who had the doublet, more complicated therapy of atezolizumab and bevacizumab, as opposed to the singlet therapy of sorafenib, experienced a longer period of time where they felt well, so a longer interval before there was deterioration in quality of life, and their symptoms really did not decline for a longer period of time while they were receiving the doublet therapy. But there are challenges to be able to scale this documentation of patient-reported data and to incorporate it into everyday oncology practice. And this is something that we've uniquely focused on here at the Dana-Farber Cancer Institute, in large part because of philanthropic support through Amy and others. So patient-reported data is not routinely collected. One may imagine that when you're meeting with your provider, they are sharing information based on the experience of other patients like yourself. But up until recently, we had not systematically collected information directly from patients to be able to integrate that with other important information in the medical record, which includes other prognostic details, such as features of your cancer, aspects of your family history, and decisions around treatment. It requires a significant technical and human lift in terms of resources required to launch and maintain such a broad system. And there is some disparity in reporting. There's disparity in access and data standards. So how do we define what is important based on what our patients are sharing? And how do we capture that in a reliable way and then map that to the other aspects of our treatment decision making? So the Dfci patient-reported data program really seeks to overcome the challenges to data collection, implementation, and use of patient-reported data with the goal of improving patient outcomes, increasing efficiency of the care that we deliver, and enabling discovery and innovation through mining of the data that is shared. This is a multidisciplinary team that, like other efforts at the Dana-Farber, requires many different and expert voices. We have a program manager, advisors, 4 technical teams that help us to move an idea from concept into production. We are live in the Yawkey Center and will be expanding to Chestnut Hill when that opens in January of 2021, and have already expanded to Merrimack Valley, which opened this year. We do have a large advisory team comprised of leadership across the Dana-Farber Cancer Institute, and I would love to highlight our patient and family advisory council members who represent both English and limited English proficient patients to really directly guide us in our efforts. Again, I'd like to show that Dana-Farber is among the most advanced in terms of harnessing the voice of the patient and integrating that information along with other key data points into our medical care. And so the National Comprehensive Cancer Network is made up of 21 comprehensive cancer centers across the U.S. And I was a part of a group who looked at how this information is being harnessed at these centers. And of the 21 centers who collect this information, only 5 centers collect it electronically, and only 2 of these centers have really put together the resources needed to integrate this within the electronic health record next to labs, next to other diagnostic information. And Dana-Farber is one of those institutions. And so this is how it works. So patients are identified based on their diagnosis, their disease center, and the type of visit, whether they're a new patient or an established patient. Patients are assigned a questionnaire, and patients then are asked to respond to the questionnaire that's appropriate for them, and they could do so using any internet-enabled device, a smartphone, a tablet, a desktop. That information then is shared immediately with the provider seeing the patient, as well as the rest of the care team. That information then could be evaluated for that individual patient at the time of the visit or between visits, or can be pulled out for aggregate analysis across a population of patients and really help us to move towards population health management so we can start to anticipate the needs of our patients and be more proactive in responding to concerns that may arise during care. We're collecting this information across the entirety of what we call the cancer care continuum, which is from prevention and risk through screening, diagnosis, those who move through treatment, survivorship, and also including end-of-life care. And so our portfolio includes a questionnaire that goes to all of our new patients at the time of a new patient visit, which includes medical history, family history, which maps to our meetings and risk stratification with the high-risk genetics team, symptoms and toxicities, and social determinants of health, which drive a lot of disparities in outcomes, such as housing needs, transportation needs, and financial concerns. We also have a tool that examines toxicities, similar to the tool that I've shared with you previously, and that has expanded out further. We are examining opioid risk tool for risk of addiction to opioids in our palliative care clinic and pain clinic. We are examining those patients who may be at risk of Lynch syndrome using a validated tool developed by Sapna Singhal. And in concert with Matt Yergelin, they have launched the Lynch Syndrome Center, which many of you have supported, and we're so grateful for that support. And this is a direct outgrowth of that, is to be able to screen all patients in the GI Cancer Center, but more broadly, we'll soon be screening across the institution to detect Lynch syndrome risk, and then refer those patients to that center for evaluation. Soon we'll launch a fitness assessment for older adults, and there are other tools that are launched as well to evaluate psychosocial distress, anxiety, depression, and some interventions around management of symptoms proactively that are coming later this year. Again, this is a testament to the support that we've received, and that we've grown the team for essentially one person to many people. And because of that support, we're able to be more proactive and more nimble in development of tools that are responsive to the needs of our patients and the providers who are meeting with patients in our clinics, and also the needs of our patients between clinic visits. So we're able to do more and be more strategic in the program's development. So I wanted to just give you a couple of examples of what that looks like for patients in various aspects of the cancer care continuum. So starting with screening and diagnosis. The new patient intake questionnaire is assigned to patients at their first consult visit in medical oncology at the Yawkey Center for Cancer Care. And it includes a broad swath of information that we need to be able to prioritize the questions that patients may have at that initial visit, and to really address their concerns quickly and be more specific in the care that we develop. We are already enhancing the tools so that the family history that is provided not only informs the care provider, but also is shared with the genetics group, so they can triage patients who may need to be seen by the genetics team more quickly and map that to their evaluations that happen within that consult. I mentioned the PREM5 Lynch syndrome screener, which has been assigned to all patients within the GCC, allows us to screen for risk of Lynch syndrome, and soon will be enhanced with the automatic referral for patients to the genetics department to enable earlier evaluation. Moving towards treatment and survivorship, the electronic patient-reported outcomes tool is a tool that is a validated instrument supported by the NCI, the National Cancer Institute, and started with an effort that I mentioned earlier, looking at common symptoms reported by patients diagnosed with cancer. Again, these are symptoms that most folks are concerned about. They want to hear what their experience might be like while going through treatment. And with this tool, we're able to give an idea to patients of what their experience could be. When may they start to experience symptoms? And more importantly, how can we be more proactive to prevent and reduce those symptoms? We have an enhanced tool that would allow us, working with our nurse navigators, to reach out to patients at the time that they're reporting symptoms, even between visits, to help them manage those symptoms, and also provide patients with tools to help them manage symptoms on their own if they're mild or moderate. And then lastly, this is a new program focused on older adult patients that are seen within the GCC. Our patient population includes 1 in 4 of our patients are age 70 or older, seen in the GCC. And these are patients who have the highest prevalence of cancer, while not the highest incidence. The numbers of patients diagnosed with cancer is certainly highest in older adults. And in the Resilience and Equity in Aging Cancer and Health Research Program, we'll be evaluating the benefit of an older adult fitness assessment to be able to determine the needs and gaps in care of older adults to really enhance eligibility for clinical trials and cancer treatments across the board, and provide the support needed to reduce unplanned emergency department and hospital visits. So what have we learned so far? So in our new patient intake questionnaire, amongst new patients, we were surprised to find that up to a quarter of our patients are reporting moderate to severe financial distress at the time that they're meeting with us for a new consult visit. And we ask patients, how difficult is it for you or your family to meet monthly expenses? And as you can see, that is a surprisingly high number. And we are beginning an effort now to examine this and other social determinants of health and match these needs to the resources available earlier in the cancer care journey for patients to meet with our financial counselors to get the help that is available. We have also learned that 40% of our patients report mental distress, anxiety, or depression at the time of their initial consult. And nearly 80% of our patients report physical distress at the time of their initial consult. And you can imagine this will help us to be able to match those patients' needs to the resources available within our pain and palliative care clinic, and also with our social work and psychosocial teams. And lastly, in examining the symptoms and toxicities that patients report as they are receiving treatment for cancer, of the nearly 2,000 patients who've participated thus far from September 2018 through August 2020, over a third of patients report moderate or severe pain while going through treatment. Over a third report moderate or severe fatigue, and just under a third report moderate or severe insomnia. All of these symptoms are quite common across the Dana-Farber, not specific to GI cancers or to the GCC, but really reflect opportunities as we see them for earlier intervention and more targeted intervention to address these symptoms and reduce the impact that they may have on someone's life and livelihood. So bringing it back to Amy, while Amy was diagnosed at a young age and faced multiple lines of therapy, she is doing quite well. She looks like she has bunny ears in the back of this picture. And in the front is a little baby, Blakely, that is now in kindergarten, doing virtual learning and struggling through that, as many of us who are parents understand. And this is Team Bushy Strong. This team has supported this effort along with others along the way and really helped us to expand and grow this program. And this year we'll be supporting our focus on social determinants of health, specifically around those distress features that I mentioned earlier. So our focus for the upcoming year is we want to continue to demonstrate the value and the benefit of continuing this effort at the Dana-Farber and nationally. We have partnerships with 2 major payers to demonstrate value-based, the value of this initiative and to help reduce costs for patients who are participating. We are focused on enhancing equity in cancer care, examining those social determinants of health like financial distress, unemployment, housing, and the like, and connect patients to those needed resources. We have started on language concordant care, making sure that patients who are not English proficient are able to participate and benefit from this program as much as our other patients. And we're starting with the Spanish language. One of our clinics at the Merrimack Valley, a third of the patient population there are Spanish speaking, and we've successfully started working with that patient group as well. And we'd love to serve additional patients across other DFCI satellites. Lastly, we're focused on improving the experience of our patients by proactively monitoring and intervening on patient symptoms. And we're doing this through a large study supported by the NCI that is led by Dr. Deborah Schrag, a colleague in GCC. And I'm a part of that research team whose focus is on reducing the impact of adverse events on interruptions to cancer care, specifically reducing unplanned emergency department visits and hospitalizations by monitoring symptoms as they occur and intervening earlier. And we are aligned with the overall strategy for digital health at Dana-Farber and the strategy for GCC to deliver compassionate care to all patients. And so I would be remiss if I did not include the full team, just so you could put some faces there to understand how many folks are required to help support this project, including our build and implementation group, analytics and data stewardship, our innovation team who helps us with a lot of the research, our advisory, and our leadership team. So thanks to all of them, and most importantly, thank you for your generous support, supporting the work that Amy, Team Bushy Strong, and many research funders have already engaged with.
Thank you, Nadine, very much for that. So I hope it gives everyone a sense going all the way from making a new mouse model and understanding the biology of some of these cancers to really deeply trying to understand and talk to our patients and use that information to help in their care. And how do you do that in a systematic way that, as Nadine shows, and has shown very nicely, really has tangible benefits for patients, whether it's symptom control or actually even living longer because they're able to manage. So it really tries to give, I think, the breadth of the work that's been done. So what we'll do then is we have a few minutes for a few more questions, and there's a bunch of questions in the question box. And then we'll wrap up. Winnie will help us wrap up, and then I think we'll then switch us over to the closing remarks from Dr. Glimcher. So of the questions in the question box, I'm going to take the first one because I think it's an important one that has come up a couple times. And it's, how does philanthropy help recruit new scientists and physicians? And I think Bob touched on this a little bit, and then Nalay mentioned it. I think the key is whenever you recruit someone new, there's a cost that's required to do that, right? So when Nalay is starting up a new lab, that's a many hundreds of thousands of dollar effort to buy new equipment, hire new people, right? Buy reagents that you need. And that has to start from somewhere. And when you're first starting, you really don't have large NIH grants and large amounts of funds to do that. So the institution has to provide that, right? Or oftentimes what happens is we as the disease center also chip into that effort. We will provide funds to make sure that the investigator comes here. Now, Nalay won't say this, but he'll also, and he did, also have many offers for many other places, right? So there's a bit of a competition to this. And some other institutions will say, well, we'll give you a little more money because we're not named quite as strong as Dana-Farber in our science, but we'll give you a little more money. Right? So we need to compete with that and make sure that it's competitive. And then I think Nadine's talk really illustrates, right, how many people it requires to impact care for patients, right? That is not a cheap endeavor. And that's someone who is dedicating her life to this effort, and as you saw, has grants that she's applied for to get this money, but needs additional money to expand what she's doing and really make the most impact to our patients she possibly can. And so that's not really recruiting new, that's sort of keeping someone here and making sure they're as productive as they possibly can be. And then I think Adam illustrates another point, which is ultimately these people get to be big and famous, right? And everyone wants Nadine to come work at their center, and everyone wants to be there. So they're going to try to take them away from us, right? And one of the ways you do that in life is by offering money to do that. And we use philanthropy for retention, right? How do we keep people here? Or even if we're sort of talking about early intervention and prevention, how do you make sure they're happy doing productive work so that even if someone calls them, they say, forget it, I'm doing well here, I don't need to leave. That's even better, right? Than getting it to the point where you need to actually try to retain them. So this money really is money we use all along the spectrum of getting someone here, keeping someone here, and making sure that they can make the maximum difference that they can for our patients. And that's really what this is all about. All right, I'm going to pick a few others and ask a couple others to answer. So this one's been coming up a lot lately. Jeff, I'm going to ask you, it's about screening for colon cancer. And it's about, in this case, the age and how there's been a lot of press about the Preventative Services Task Force changing guidelines. How has Dana-Farber been working in that space and some of the young onset work maybe that you and Kimmy are doing?
Sure, yeah. So, and for those who were here last year, I think Kimmy talked about young onset, this whole concept that the incidence in older adults has relatively gone down. It's still a more common disease, colorectal cancer, people greater than age 50 in their 60s and 70s. But there's a steady rise in people under the age of 50. And the question is what to do about that, both in terms of why So Kimmy has created a center, and we actually have applied for funding to collaborate with other institutions across the country to collect a large cohort of patients to understand why there is a difference in the incidence in patients who are younger. And then the question is what to do about in terms of screening. The American Cancer Society came out pretty early on, several years ago, suggesting age 45. Others haven't. I actually just saw, technically, actually, the U.S. Preventive Service Task Force, which is sort of the body that sort of makes usually very conservative recommendations in terms of screening, in terms of thinking about lowering the age of initial screening at 45. It's actually a preliminary report. It's actually not a firm recommendation. It hasn't been definitively approved, and they're sort of getting feedback right now in terms of other stakeholders thinking about that. But, I think, again, the importance of, trying to think about it earlier on, as important is really just education, because what many groups have found is that younger people will have symptoms that often aren't colorectal cancer related, but they're also ignored because they're assumed they're not related to colorectal. So much earlier thinking if you're having bleeding or abdominal pain or changes in bowel habits, to not ignore those and to really bring them up with your physician.
Thanks, Jeff. Jen, I'm going to ask you a question. One of the questions is, some of the cancers that we treat are less common. And are there barriers that you find to doing research in tumors that are less common? I ask you, obviously, because of your tremendous expertise in neuroendocrine tumors and leading that program. How does that influence how you lead that program and the work you can do?
Yeah, I think that across the board, there are challenges with rare diseases. And I think there are certainly particular challenges to neuroendocrine tumors. Some of it even extends to, for instance, work like what Nalay has been doing with models. This is a disease, neuroendocrine tumors, where we really have not had really great cell models. So there is work that is being done. We're trying to collaborate with investigators at the Broad to create these models so that we can replicate some of the work that has really helped with the basic understanding of disease. There also are inherent challenges to studying a rare disease when it comes to clinical trials. It just takes longer to, recruit patients when you have a rare disease. And it really has forced us to collaborate with other institutions to try to get the work done.
Great. Thank you, Jen. There's a question about, with all the different diseases that we deal with, how do we balance with such a large faculty where we put our emphasis? Maybe, Bob, I can ask you, in your time doing this, how have you thought about bigger picture things like that?
Well, as you pointed out in one of your early slides, there are more than 10 different cancers that comprise the GCC, and many of them have very different backgrounds and very different volumes and very different causes and very different outcomes. I think we have an obligation to be treating and caring for people who come in our door, but we also have an obligation to take more unusual problems, such as Jen was just talking about, but cancers like of the appendix or of the anus or of the bile ducts, which are not the most frequently identified cancers of the GI tract, and becoming a repository, a place that people can come and participate in clinical trials, learn about new approaches, and be cared for in the most manner.
Yeah, thank you, Bob. I fully agree, and I think really the motto we've taken in the GCC is we form teams of smart people. I think ultimately that's what gets the job done, is when we have a team of smart people focused on a particular problem. And we try to make sure, and we look very frequently at who are the people working on different diseases, how do we make sure we're interacting, and then how do we have a coherent plan across our teams. And so this is something we sort of continually reevaluate. And I think, or at least I hope, you can see in that the people we hire, we try to make sure fit niches that we need to advance the team, to make the team more successful, to make the advances more prominent and more rapid. And I think that was leading me to another question, which maybe I'll ask, I'll start off asking Nalay. So it's asking about the rapidity of movement between sort of the lab and the clinic. And let me ask you, as you've been thinking about some of this work, what strategies have you taken to think through, well, I'm finding this in the lab. How do I make sure I'm making it as speedy to the clinic as it can be?
Yeah. So, I mean, how do I think about translate? Oh, am I?
Yeah, you're a little quiet.
Okay, sorry. Okay, I hope I sound better now. Okay?
A little louder.
How about now?
Why don't you fix, and maybe I'll pass the question to another person, and then you can get that to be a little bit louder. So maybe Nadine, I'll pass that question to you. You know, I think what you have done with the patient-reported data program is quite amazing, and I think as you described, this is not something that even other major cancer centers have been able to do. How do you think you made that happen, going from an idea that you wanted to implement this to actual its implementation, and what were the things along the way that either helped you or stalled you?
Certainly was having support to explore the idea was the most important. Secondly, the motivation to do that. And I think thirdly, having very strong rationale that this was in fact going to help our patients and be beneficial. I think it's always the reason why you would want to invest this effort. You know, the Dana-Farber, along with our colleague hospital healthcare system, really invested a lot in our medical record system. And we saw that there was a gap in terms of the data that we were collecting, and we needed to really enrich all of our diagnostic studies, all of our imaging studies, all of our treatment data with the experience of the patient. And seeing that the FDA supported that for drug approvals, and knowing this was going to be incorporated even more commonly in clinical trials, we wanted to make sure that we were ready and that we had a system in place that would work for our needs moving forward.
Thank you.
Brian, I wonder if you could pick up on the question that you posed before with the use of matching genomic information with clinical trials. If one is going to think about taking something from the clinic to the lab and back to the clinic, to me, that's the most contemporary utilization of it, and your work in pancreas cancer certainly represents that.
Yeah, so thanks, Bob. So I think from the way point that as in the lab, we identify either single alterations, so changes in a specific gene, or patterns of alterations that go together that can direct us to therapies. That is key for us then in the clinic to identify. And we need to find those people because we now have a hypothesis that they could benefit from a particular therapy. And I think the other thing that Dana-Farber has really invested in is making sure that every patient that comes here has the opportunity to have their tumor sequence, right, the DNA in their tumor sequence. With that, we can then find the patients that Nalay was talking about, right, that had a P53 mutation, that had loss of P16, and find these combinations. And I think what Bob is referring to is that we've, within the GCC, heavily invested in that machinery. And we actually have a specific group, and that group is actually led by Marios Giannakis, another one of the medical oncologists, to make sure that we have a screening program that every patient who tumor gets sequenced, we look at them centrally by a committee, and we pick out those ones that fit these specific types that we think would benefit from a particular therapy. And therefore, it does not rely on every single doc to go through every little genetic change and make a decision. It's actually a centralized process that then helps us do this. And that matching process, it really is what I hope facilitates then the most up-to-date therapy choices for our patients. So I think that is, as Bob is alluding to, another way to make sure that the latest science goes into the clinic. And that's really our real goal here, is take the latest science going on and make sure our patients benefit from that. And then beyond Dana-Farber, ultimately make sure the world benefits from that. Right? Really that we extend that beyond us. All right, so I'm starting to get the NGEL warnings that we need to wrap up because we're starting to run out of time. I think we could talk for a lot longer, and I think all of us on this video would love to do so. So we will follow up and find other questions we can talk about and things we can do together. Why don't I pass this to Winnie to help us give sort of a tie-up for this lovely event and some insights that she may have reached listening to this, and then I'll just say our final goodbye.
Well, first of all, I want to thank the faculty members for taking the time out to prepare these presentations. I think you've clearly illustrated how Dana-Farber, as the Institute, approaches our fight against cancer. You have basic science research on the one hand, which brings new medicines forward. And then you have the program described by Nadine, which covers compassionate patient care that is beyond the prescription of medicines. So truly, this is an all-encompassing and very compassionate approach. And I hope the audience also appreciates that and how your support enables all that we do here. So I thank you all for taking the time out to listen to us. I hope you've enjoyed listening to the sessions this morning. Enjoy your yoga stretch and the piano interludes. And I also thank Nadine, Nadine and Lei for putting the presentations together, and Angel and her team for putting so much effort into orchestrating this whole event, which is very difficult because we are experimenting with new technology platforms and all of us have new passwords and trying to find what buttons to use to get ourselves on camera. So it's been quite a challenge. And Brian, of course, thank you so much for your leadership. And we really, really appreciate all that you have done.
Well, thank you, Winnie. I mean, really just to thank all the people who, joined us for this event. I really look forward to meeting the new members of our committee in person. I very much hope that is next year or in between if opportunities arise. And also really a very large thank you to the people who come to this each year and have really been such important supporters of the GCC. I think really highlighting what Winnie said, so much of what we do requires support. Again, moral support and financial support and vision support. And you all provide that to us. And we use that. And I mean, really, I think you can probably get the sense this is our lives, right? This is our passions. This is how we spend huge amounts of our time thinking about and wanting to improve the lives of patients. And you help us get there. And we know next year and 2 years from now and 5 years from now, things will be even better and substantially better than they are today. And you all are the vehicle that helps us do that. So thank you so much for joining us. And again, her efforts in organizing the event and really helping us with the vision for what it should be, and to Angel and her team. And then certainly to the faculty who joined us and shared their research with us. So I think next up is the closing remarks from Dr. Glimcher, and we will, with this and a last thanks, close the GCC event. So thank you, everyone.
I do have one last comment. We're looking at the last quarter here. For those of you who are planning your gifts, please keep in mind that the CARES Act has special provisions for the year 2020 only, that you get special tax deductions for your charitable gifts. So please get in touch with your tax consultants and think about your gifts planning for the rest of the year. Thank you again for your support.
Bye, everyone. Thank you.
Hello again. I hope you all enjoyed a productive breakout session and the time to connect with one another. Please accept my sincere gratitude for your participation. And again, I would like to thank Dana-Farber's trustee and faculty chairs whose names you'll see on your screen for leading today's discussions. They have diligently taken note of your questions and ideas, and I'm looking forward to meeting with them next week for an executive session to ensure that I am apprised of your questions and your ideas and your feedback. While, of course, we would have preferred to meet in person, as we have for nearly 2 decades for this symposium, one distinct opportunity of this year's format is the ability to expand access to this program to an even wider community. In fact, at the executive session last year, one of the main recommendations from our trustee and faculty co-chairs, based on your feedback, was to shape share this symposium more widely through video and technology to enable even greater participation for those who might not be able to get to Boston. To that end, recordings from today will be available to you and to those who couldn't join us live. As well, the community and resources sections at the top of this event platform will continue to be available in the months ahead, and those are additional ways to stay engaged with each other and to spread the word to others about Dana-Farber's advances in research and care.
Jimmy is a boy, saved from leukemia by Dr. Sydney Farber. Jimmy is a girl, a nephew, a husband, a wife. Jimmy is every patient ever at Dana-Farber Cancer Institute, their families, friends, neighbors, every doctor, nurse, and researcher at Dana-Farber, every event walker, runner, rider, and golfer. Support the Jimmy Fund and give more and more of us a second chance at life, because we're all Jimmy.